Welcome to the Chemistry, Biology and Math Revision Hub. Today we're doing the A-level biology topics, and the topic is ecology. For topic 5B1, what is ecology? Of course, we're going to be looking at the different definitions. In ecology, ecosystems, these are all living organisms and the way they interact with each other, as well as with a non-living part of the environment. A habitat is a place where an organism lives, and a population is a group of organisms of the same species living and breeding together in a specific habitat. A community includes all populations of the different species of organisms in a habitat at any one time, so they have to be in the same location. A niche, this is the role of an organism in an ecosystem. So these definitions are really important. And then when talking about ecosystems, we have to look at biotic and abiotic factors. Biotic factors are the living elements of an ecosystem, and these include predation, disease, finding a mate, and so on, while the abiotic factors are the non-living components of a habitat. So these include maybe weather, climate, temperature, sunlight, soil factors, as well as oxygen availability, and so on, depending on the needs of the organisms surviving in that area. And looking at the major ecosystems, we have already looked at all these definitions. Lastly, we're looking at the biome and the biosphere. Biosphere are all areas of the surface of the earth where living organisms survive, so you can see it's bigger than a biome. Biomes are all major ecosystems of the world. In topic 5B2, how ecosystems evolve, we're going to look at succession, and this involves primary as well as secondary succession. Succession is a process by which communities of plants and animals colonize certain areas over periods of time, and those are going to be replaced by others. Usually those others are going to be more advanced, and then they'll produce communities. For succession to occur, we need pioneer species. These are species that colonize the area initially, or they are part of the first stage of succession. They colonize the area. The area could be a bare rock, it could be sand, it could be clay, it could be on ice. These organisms are usually called opportunists. Examples are algae, mosses, and fungi. They can penetrate the rock or any surface in various ways. For example, if it's a rock, they could dissolve it with the acid they secrete, and then penetrating the cracks with root hairs and hyphae. They should be 1S. So this is going to break down the rock, trapping organic matter when they die, and then this is going to generate humus, and the humus is going to be the first part of formation of the soil. When we talk about succession, there is usually a climax community. This is a community that remains generally constant over time. It's kind of going to be stable if there is no intervention. A plagioclimax community, this is a subclimax community where succession has been held back by human activity. So this is an example of primary succession where we see a bare rock colonized by pioneer species. This could be algae or moss. These are going to change the environment, and when they change, the conditions will no longer be conducive for them to survive. So they're going to die, and when they die, decomposition is going to occur, and this is going to create humus, leading to the first part of the soil that is going to be formed. And the changed conditions are going to allow for more advanced species to come and colonize the area, and over time, we see a climax community is going to be maintained. Secondary succession occurs when there was initially a climax community that is going to be destabilized. For example, it could be a fire, it could be a volcanic eruption, or anything that changes or destroys organisms that were part of the climax community. Even with this great destruction, though some seeds are still present and there is soil, secondary succession is going to occur faster, and eventually a climax community is going to be re-established. If we compare secondary and primary succession, this occurs in area where no life existed, where this occurs where life existed but was destroyed. This is going to be slower, this is going to be faster because there is already soil. This occurs in areas which could be a bare rock, a barren island, ice, as well as cold lava, but this usually occurs in abandoned farmlands, burnt and cut forests, and so on. Topic 5B3, effect of abiotic factors on populations. Abiotic factors are the non-living components of an ecosystem. This could be climate-related, soil-related, or maybe oxygen-related or other factors. So for climate-related, there is temperature. This controls how enzyme activity occurs. Of course, the higher the temperature, the higher the enzyme activity until the optimum, and after that, enzymes become denatured, and so the rate is going to decrease. There is sunlight. Sunlight is going to control how fast photosynthesis occurs. Rainfall, of course, this provides water. This is also going to control how plants do thrive within an area, or even how organisms thrive within an area. Because in some areas that are kind of dry, rainfall provides the water that the animals need to drink. Rainfall provides or waters the soil so that more things grow. So this is going to affect the abundance and distribution. There is wind and water currents. For example, this could be hurricanes and tornadoes, and these have a way of affecting the distribution as well as abundance of organisms within an area. When we go to edaphic factors, which are soil factors, loose soil and shifting soil allows less growth because, of course, the roots are not going to be anchored properly and the nutrients are going to be washed out easily. The amount of water within the soil can affect how abundant organisms are, the pH of the soil, because, of course, this affects the microorganisms and how nutrients can be recycled. The mineral content, this is also important because plants need minerals, and even organisms that thrive within the soil need minerals in order to survive. When we go to oxygen availability, cold and fast-flowing water contains more oxygen or more dissolved oxygen, while if the oxygen concentration is lower, then the survival of organisms is going to be affected, whether in the soil or in the water. Moving on to topic 5b4, effects of biotic factors on populations. So here we're going to look at predation, finding a mate, territory, parasitism, and disease. For predation, the more the predators, the lower the number of the prey. You can look at some case studies in your textbook. Finding a mate, if the organisms are abundant in an area, then it's going to be easy to find a mate and they will reproduce sexually. And this is going to increase the offsprings or the number of organisms within that area. For territory, the type and size of territory will determine which species live in a particular area. If it's small, if it's desert, and so on, that is going to determine which organisms thrive, meaning those that are better adapted for those conditions are going to thrive in that area. For parasitism and disease, more parasites and diseases affect the growth of organisms, and the parasite and diseases usually spread in areas where organisms are densely populated. In topic 5b5, interactions and the importance of the niche, we're going to look at distribution of organisms. Of course, there's some case studies in your textbook, but you can look at that. Survival and distribution of organisms is sometimes affected by factors. This could be density dependent or density independent. The density dependent factors affect organisms that live in a certain area. These include parasitism, competition, and disease. If there are more organisms within an area, then the parasites are going to spread really fast. There is going to be greater competition for resources, and there is going to be faster spread of disease. For the density independent factors, these affect the number of organisms in a certain area or certain niche, regardless of the population size. Of course, these include temperature. Within that area, it's going to be the same temperature, no matter if it's densely populated or not. Natural disasters are going to affect all organisms the same way. The level of oxygen in the atmosphere is going to affect them the same way, whether they are densely populated or not. When we look at competition, there are two types. There is intraspecific competition, meaning within members of the same species, and the other is interspecific competition, meaning between members of different species. Competition could be for food, it could be for territory as well as mates for those in the same species, while interspecific competition could be usually for resources. Of course, these resources are going to be food, they're going to be water, and so on, as well as territory, but they will not be fighting for mates. In topic 5b6, investigating abundance and distribution of organisms. Looking at abundance and distribution, abundance relates to the number of organisms of a particular species relative to other organisms in the same habitat. Distribution is to do with where these organisms are found within the environment. Some organisms can be randomly distributed, meaning they are arranged in no pattern. That's why we call it random distribution. This occurs usually when there is no hostility and there are plenty of resources. Others are going to be uniformly distributed or evenly spaced. This occurs when there is thin distribution of resources or hostility present among individuals of a species. So they want to be spaced to avoid fighting or to avoid problems. The last one is called clump distribution. This could be common in herds. Animals could converge or gather where resources are in order to have enough to eat or enough resources. Moving on to ecological techniques, the first one is looking at quadrats. We have 4 types of quadrats. We have frame quadrats, point quadrats, permanent quadrats, as well as area quadrats. From quadrats could be of various sizes, and these are going to be thrown randomly like you see here. Of course, you count the number of organisms per quadrat and then repeat the throws in order to calculate the mean, and then you can use that to scale up. The second one, we look at the point quadrat. In this type of quadrat, metal pins are going to be dropped through each hole. Like we see here, we have this quadrat with so many holes as we see there. The metal grade is going to be thrown through each hole, and then number of organisms that are hit or touched are going to be recorded. Here we can calculate the percentage cover by saying number of hits divided by the total number of pins times 100. Number 3, we go to the permanent quadrats. These permanent quadrats are going to be left in place at all time for longer periods of time, and data is going to be corrected for seasons throughout the year in the same place. So it enables you to compare what you saw in the spring to the winter and other seasons of the year. Area quadrats are high-tech, and these are take area photographs of large areas of land, and those areas are going to be continually surveyed for numbers of species in order to determine the abundance and distribution. Going to the second ecological technique, we're going to look at transects. There are two types of transects. They are line transects as well as belt transects. Line transects are not random. The tape is going to be stretched at 2 points, like we see this point and that point. And then we record the individual plants or animals that touch the tape. And then for the belt transect, here we can use two tapes, stretched at ground level, and then quadrats could be placed, like you see this one here, at regular intervals, and then the number of organisms within each quadrat are going to be counted. And again here, you're looking for specific organisms, so you count the number of that specific species within an area of that quadrat, and then you can find ways of scaling up to determine the total number of organisms within a specific area. For these ecological experiments, usually they're going to ask you to measure abiotic factors. For example, if you're studying the effect of temperature on abundance and distribution, how are you going to measure the temperature? You need to know. This could be temperature of the air, temperature of the water or the soil. You could use a glass or digital thermometer. For light intensity, we use light meters. For pH, we can use pH indicators, but pH meters are better. For wind speed, we use anemometers. For slope of an incline, we use clinometers, and then oxygen levels in water, we use a digital oxygen probe. So for any experiments requiring you to measure the abiotic factors, you could use these methods to measure those abiotic factors. So topic 5b7, statistics and ecology. So here are the steps we have to follow in any statistical test. We have to state the null hypothesis. Then we have to decide which statistical test we're going to use. We have to carry out the statistical test, and then the critical data is going to be at the value of p is equal to 0.05. Then we will compare the statistical value as well as the critical value. This is obtained from the table of critical results. And then we can make a conclusion on whether to reject or accept the null hypothesis. The null hypothesis is always going to begin with no significant difference or no correlation. The critical value in biology is going to be at 0.05. We accept results if there is a 5% of results are by chance. First, we're going to look at Spearman's rank correlation. This is used to see strength of correlation between two sets of continuous data measured from the same sample. We are going to assign a rank to all the results. If the result is +1, -1, or 0, then we find how significant it is. Here, the null hypothesis should be there is no correlation between the two data sets. And we use this here to calculate Spearman's rank correlation, whereby n is going to be the number of pairs, and d is going to be the difference between the data sets. Like we said already, the value is going to be between +1 and -1. If we get a +1, it's going to be a perfect positive correlation. And if we get a -1, it's going to be perfect negative correlation. If it's 0, there is no correlation. So of course, the results are going to be within this range. If it's closer to 1, then there is greater positive correlation and so on. Let's look at an example from the textbook. This is the formation of a new volcanic South island. We have a table of results. We have the years after formation as well as the species total. So this is the x value, which is the number of species. We have 0, we have 4, we have 10, 13 until 54. And then this is the rank for x. So we are going to arrange them in increasing order. Then these ones here, the y values are going to be the years after formation. Still, we have 0 to 40. And again, they're already arranged in increasing order. We have 0, 5, 10, 15, 20, 25, 30, 35. Again, 1 to 9. So we're going to get a difference between the ranks. That minus that gives us a 0. that minus that gives us a 0, that minus that we get a 0, but that minus that gives us a -1, and then that minus that gives us a 1, and so on. So we see the rest are going to be 0, but we have a 1 and -1, meaning if we do the d squared, it's going to be 0 squared is 0, 0 squared is 0, that squared is 0, -1 squared is 1, and +1 squared is 1. The rest are going to be 0. When we sum up all these to get the total of the d squared, we're going to get a 2. Now remember our formula for calculating the rank correlation is going to be 1 minus 6 times the summation of d squared. So this is going to be 1 minus 6 times 2 divided by the number of data sets is 9. So we have 9 into 9 squared, which is 81 minus 1. So it's going to be 9 times 80, and this is what results. So using your calculator, you're going to get 1 minus 0.0167, which is 0.983. When we look at the table of critical results, we can see at n number of pairs equal to 9, the critical value is 0.68, which is less than the 0.983. So we reject the null hypothesis. The next part is looking at the student t-test, which is the second method of statistical analysis. This is used to find the difference between two sets or two means from different samples. Here we can find whether the difference is significant or not. If the t-test is less or greater than the critical value, we can make a conclusion whether to accept or reject the null hypothesis. Here we have to find the mean. This could be mean of the first data or mean of the second data. So x bar is equal to the total or the sum of x divided by n. And then this one here is going to be sum of x squared minus the sum of x bracket squared divided by n minus 1. And later we can use that here to find the t-value. The terms used here are defined here. Let's look at the example, the shells from two beaches. The null hypothesis here is there is no significant difference between the length of the mussels from the two beaches. So from the table, we're given the data. First, these are the x values, or let me call it the x1 or the first data set, the value of length of the largest from beach 1, and there is a second beach. So I'm going to get this x value and square it. This squared gives us that squared gives us that. So basically squaring all these values until here is going to give me what we see here. And in the end, I'm going to add up all these squared values to give me that. This is going to be the summation of x1 squared. Then I'll do the same thing here, square this to give me that until there, and then add up to get this. So remember, we have two different formulae we have to use. I need to find the mean for both data sets. So the mean is going to be this total divided by the number of items, which is 10. So it gave me 74.9. And the mean for this second data set is going to be 768 divided by 10, which is 76.8. Then I need to substitute to find the S1 squared. And again, remember, we use this. So this is going to be the summation, which is 56, this one here, 873, minus that squared divided by the total number, which is 10, divided by n minus 1, which is going to be 10 minus 1, giving us a 9. And when you calculate, you're going to get 85.88. For the second data, of course, we're going to do the summation of x2 squared, which is going to be that, minus the summation of x2, which is going to be 768. We square that and then divide by the value n, which is 10. Divide everything by n minus 1, which is 10 minus 1, giving us a 9. So the answer is going to be that. Remember, n1 and n2 is 10. So when you substitute into the formula, you're going to get n minus 1, which is 10 minus 1, giving us a 9. And now that we have this value, that value, this value, and that value, we can substitute it into the final formula to find the value for t. Our t is going to be the mean for the first data, which is that, minus the mean for the second data, which is that. Divide everything by the square root of s1 squared from the first data. Divide by n, which is 10, plus the s2 squared of the second data, divide by n, which is 10. And using the calculator, it gave us negative 0.57 to 2 decimal places. For the t-test, we need to use the degree of freedom. Degree of freedom is going to be the sum of two values of n minus 2. In this case, it's going to be 20 minus 2, which is 18. We go to 18 degree of freedom at p is equal to 0.05, and this is going to be the value we find. The calculated value is going to be lower. Remember, our calculated value is 0.57. Because our calculated value is less than the critical value, we are going to accept the null hypothesis. Lastly, we'll look at the chi-square test. This is used to compare two data sets, meaning the observed as well as the expected. So there is a theoretical data set, and then there is the actual result from the experimentation. These are going to be great in genetics as well as in fieldwork sampling or fieldwork studies. When we use the chi-square test, the null hypothesis should be there is no significant difference between the observed and the expected frequencies. And the formula is going to be x squared is equal to the summation of the observed minus the expected squared divided by the expected. And we'll do this at degree of freedom equal to n minus 1. The results we have here, we see regions where both clams and oysters are present, they are 7. Clams are absent, but oysters are present, they are 15. So when you add up, we get 22. And then areas where the pearl oysters are absent, but clams are present, are 20. Where the clams and oysters are absent, we see 8. Adding up, we get 28. So the sum of these, we get 27. The sum of that, we get 23. The sum of that, we get 50. And of course, we've already summed up like that to get 22 and 28. So this data here was calculated by multiplying the 22 here, the sum here, times the sum there, divide by the total, which is going to be 50. So we get 22 times 27 divided by 50. For the absent, we're going to get the sum here, which is 22 times 23 here, divided by 50. And then for here, we're going to get the sum here, 28 times 27 divided by 50. And for this data here, we're going to get 28 times 23 divided by 50. And when we add up that plus that, we get 23. Adding up that plus that, we get 27. And then adding up here, we get 22. Adding up there, we get 28. And of course, the total is going to be 50. Remember, the degree of freedom should be n minus 1. And since here we have two data, we're going to be 2 minus 1, which is 1. The observed, we see 7, we see 15, we see 20, and we see 8 from this table here. And then the expected from this calculated table here, we see 11.88, we see 10.12, and then here we see 15.12, which is that, and 12.88, which is that. The observed minus expected squared for this part gives us that. Then observed minus expected squared gives us that. When we divide by expected, we get 2, and we get that. Then here, observed minus expected squared, we get that. Observed minus expected squared, we get that. And then dividing by expected, we get that and that. When we add up everything, meaning adding up that, adding up this and this, we get about 7.78. When we go to the table of critical results at degree of freedom equal to 1, at p is equal to 0.05, this is going to be the value we get. And we can see our calculated value, which is about 7.78, is greater than the critical value. So we're going to reject the null hypothesis, meaning there is a significant difference between the two data sets. And again, if you want to practice more with these statistical tests, you can check out the Unit 6 papers for biology that I've done on my channel so that you can be able to see more actual practice exam questions. So this brings us to the end of this topic. Thank you for being with us. Do not forget to subscribe. See you in the next video. Bye-bye.